Welcome to our BovReg final conference. I'm really happy that everybody could make it either here on site or all on the different laptops, computers, PC screens online, and hopefully we will have a very nice and informative conference for all. Whoops. This here. It's very, very sensitive device here. So a few housekeeping announcements. So this is a fully hybrid meeting. So you will have the chance to stay tuned here online and also on site. All the presentations will be recorded and will be available after the conference. So no rush. If you want to reread or look at something after the conference, you will be able to do so. For the conference attendees, we want to really have a good representation of yours in the discussion. So by default, you're muted. But if you want to discuss, have a comment or a question, please use the Zoom indicator, raise your hand, and then your muting will be released and you can take part in the discussion. In case of trouble, please use the chat. We have here colleagues who will take care of that. For the audience and online, please speak loud and not too fast. And for the questions raised here in the audience, please use the microphone and give your name so that people online can know who you are, who's asking the question, and also can understand. If you just ask your questions in the audience here, the online attendees will not be able to hear. And finally, for the on-site attendees, they have the advantage that refreshments and everything will be provided in the hall. This is really... I have to... yeah. So probably you know that this conference here is a beat in the chain of the FANG anniversary celebration. So FANG, the Functional Annotation of Farmed Animal Genomes, celebrates its 10th birthday. So this is what we are celebrating, and BovReg is part of this FANG community. So we are part of the celebration too. Everything starts for BovReg and also for FANG with the Ag Encode workshop in 2014. And in 2015, there was this white paper. And for those of you in the audience who are not very familiar with FANG, I think the ones here online on site can take a rest for 2 minutes. So it's a non-institutional organization because we don't have the opportunity like the Human ENCODE project having this NIH big spending funder in the background. So people decided that they had to do this voluntarily and you subscribe simply by indicating a membership. The dedication is that we want to make more of the DNA alphabet of the online genomes which are available and give some meaning to them by functional annotation. Currently we have about 500 contributors, but early on we discovered that voluntary actions are nice, but most of the times they lack money and they also lack coordination. So it's a kind of a little bit of a history for the particular European part of FANG, starting in 2014, when people decided, okay, we need some kind of a structure. So a Coast Action Eurofang was initiated, and successfully a community was established, which then led to communities which could then reply to calls for Horizon 2020 projects. So 3 initially projects were funded and another 6 were substituted later on. So we have the Eurofang cluster from those Horizon 2020 funded projects. And quite recently we have the Eurofang research infrastructure, where we hear more about it on the next day. So about coming back to BovReg, we started the work actually, I remember when I was at I think it was the ISAC in 2017, when the first thoughts about BovReg were initiated. And then the EU launched the Horizon 2020 program, indicating that there is more there's more need for understanding the biology behind genetic variation. And so we contacted people, asked them to participate, and had 2 preparatory meetings in Amsterdam and Paris to submit our proposal. And finally, we got funded. So BovReg, what is it about? It's EU-funded, Horizon 2020 funded consortium within the global FANG, and we have a focus on biology-informed breeding. Our aim is that we want to generate functional genome data and subsequent also standardized data analysis with a catalog of FANG assays, which we had agreed upon in the global FANG community, to identify functionally active genomic features which contribute to the phenotypic diversity and plasticity of the bovine cattle. So it's about Bos taurus. We don't deal with Bos indicus. We have right from the beginning 3 target traits, which are robustness, namely fertility, which is health. There, our example is mastitis, and then also we address biological efficiency, which is production and feed efficiency. We started in November 2019, and initially we planned to end by August 2023, and then something came in between, something very tiny but very meaningful, as you can see here. So in February 2024, now in 2 weeks to come, is the end of the project. The virus which came between us, I know more about viruses now much more than I had anticipated several months ago, but the consequences were quite heavy, so we had really specific challenges to work under COVID SARS-CoV-2 conditions. We learned how to deal with Zoom conferences. I think we wouldn't have planned to have so many conferences if we hadn't had COVID and the option and or the need to go online. We worked with masks, and several of our colleagues, for example, they had to set up databases for SARS-CoV-2 sequences. They even had to do the routine SARS-CoV-2 testing, which all caused a substantial delay of our project. But nevertheless, I think we really, with the dedicated action of many colleagues, we managed to overcome all these issues and could set out to achieve our objective, which were, we wanted to establish new laboratory and bioinformatic tools. We wanted to annotate the functionally active genomic regions. We wanted to map functional parameters, which range from phenotypes, from transcriptome to zootechnical phenotypes. And we also want to establish prototypes, how this biological knowledge could be implemented into genomic selection processes. And finally, last but not least, we also wanted to address how to disseminate this knowledge and then assess also how society might perceive how we deal with the new opportunities, maybe in parallel to developing those opportunities. Our consortium comprises 20 partners from 13 countries and 2 international institutions. Luckily, we are even able to have partners from Australia and officially even Canadian partner there, which I think is a really, at that time, I think it was one of the very, very few officially approved partners as Canada. Now things further improve, I think, but at that time, it was really something unique. And the setup of the project is more or less, we had essentially 3 areas. One is to establish tools and resources. The other is to really function and to provide the background and the devices which we need for functional annotation, do the functional annotation itself, which is in beige, and then distribute knowledge and exploit this information for genomic prediction. And finally, in the black bar, how do we distribute this across the community, to stakeholders, politicians, and society? We have BovReg work is distributed in 10 different work packages. One work package on development of laboratory tools and resources. One with the lead Dominique Rochard from INRA. Sorry for deleting this E. Then we have the functional and structural genome annotation with the team in GIGA, Carol and Gabriel in lead. We have the tools, data, and pipelines hub for bioinformatic analysis, the team from CRG with Cedric and Jose. Then we have the integrative genome phenotype data analysis with Hubert from EDH. We have the epigenetics and environmental impact work package with Jens Wanselow from FBN. Functional validation of regulatory variants from Hiroaki from Poland. We have then one work package on biology-driven selection with now the leads from Emre and Birgit. Then the societal ethics and societal issues work package with Frank and Köln-Kramer. Finally, training, communication, dissemination, which is an essential part of all EU projects with Johanna and Terhi, and finally also the BovReg FANG and Data Coordination Center. You see underlined, we have had a particular interest to also recruit young scientists, underlined here, to give them responsibilities in the work packages, and I think it worked quite well. Many of them really prepared for taking over new responsibilities in the next years. For example, also Hubert, I congratulate him. I don't know if he's in the audience now for this fixed position at the ETH. It's really a very well-known and renowned position. So I think we were also quite effective, I think, in promoting young scientists. So the implementation, as I mentioned, has several areas, and although the work is organized in work packages which are more relevant to the different disciplines, the different implementation areas, they work across different work packages, and this is something which I would like to explain in the next minutes. When I prepared for this talk, first of all, I thought, oh wow, it's 1 hour, it's so much time, how do I spend this? And I don't want to be like Leonid Brezhnev or Mao Zedong talking in the audience for hours. But in the end, then when I looked at all the different things which have been done in the last 4 years, I discovered, 1 hour might be way too less time to do so. On the other hand, I had the challenge that we have many very, very dedicated and interesting speakers in the next 2 days. So I don't want to do the cherry picking. I want to give the credit to them. They want to present their results, their achievements. So I will just give an overview and give you some kind of a philosophy which we have across the different work packages and the different objectives. So first of all, we wanted to establish laboratory and bioinformatic tools. Laboratory tools. So we want to, on the one hand, develop new laboratory resources. And for this, when we started the project, we thought that in vitro models are in desperate need in our communities. We have some in vitro models which we work on already for decades without much knowing about them. So there was a need to develop new cell models, and entirely new. And on the other hand, as I mentioned, we are working sometimes for decades with established cell models, taking for granted their properties, their efficiency, and this is probably something which is not very much to the point. And we discovered several surprises when we worked on those cell lines. For example, we looked at their genomic composition and also on their expression level. This is one thing which will be presented by 3 talks. One on presenting the work on the existing cell lines, then one talk about establishing a new cell model for granulosa, for ovary cell work, and then another one for establishing new cell lines for working on liver cells, which I think especially in the bovine community is in desperate need. So that's about developing new laboratory tools and resources. The second one is no, that's one too far. Yeah. Then on the other hand, let's go one back. Yes. The other one is resources. When you have data, you need to do some. Or if you have raw data, and nowadays our omics technologies release huge amounts of data, we would need then pipelines. And from the beginning, we ask ourselves which pipelines are needed to process and provide all the standardized output we would like to have from all our assays we conduct. So first of all, we started with surveys to explore the needs within our community. And second, then when we had explored the needs, it was quite an anonymous or we all agreed that definitely we need something which is standardized, which can be applied across different computing environments. And I think the colleagues from CRG, they easily persuaded us that NF Core Nextflow would be the best choice to do so. And I think they were even so persuasive that these things were also adopted across other projects. And we will hear more about this in a talk from Jose from CRG. And I think it also has, even within the NF Core community, we made an impact, and this has consequences, and he will definitely show how this worked. And I think this is also a legacy which we provide beyond the cell models. It's also something which we provide to the entire global FANG community. Then, when you have data and when you have analysis, you need some time, some place where to deposit all this. So another legacy is the BovReg data, not the BovReg, the FANG Data Coordination Center as a data portal. And the data portal initially just was a kind of a drop of the different data sets, but it further evolved. For example, in BovReg specifically, we realized that most of the times when people want to recruit and merge data from different sources for their secondary or tertiary analyses, they need ontologies. So BovReg particularly contributed to the Data Coordination Center by efforts creating on new ontologies, particularly, for example, when we worked with new cell models, when we worked with embryos, different environmental stages. This created the need for new ontologies, which are definitely distinct and new compared to the human communities. So if you nowadays look into the data portal, you will see data sets, and you will also see pipelines attached to the data sets, all for downloading. And for this, we will have an extensive session informing about the bioinformatic tools and databases from Peter Harrison from Amble. Hopefully, his train isn't delayed, so we may make it just in time for his presentation. So nowadays, when you look, and this is maybe particularly for those people in the audience who are not really very familiar with FANG and BovReg, this Data Coordination Center, which has contributions not just from this work package 10, but all the input there comes from the work packages 1, 2, and 5, at least from the BovReg side. I think it's a treasure trove. Even if you are not interested in the BovReg data per se, there is, for example, a lot of protocols. For example, if you want to do single cell, if you want to do RNA sequencing, if you want to do whole genome bisulfite sequencing, just look there for the protocols. They are very, very, very detailed because this is a demand of FANG. I think these details go way beyond usual protocols you find in papers or you find in some instructions from the suppliers. So this is all presented there. So it's about the data itself. It's the metadata, which are very, very rich. It goes down to even the latitude of where the sample is collected, and also the distribution of methods I mentioned. And all this is in a structured way, fitting into the Amble EBI environment with biosamples ID, with INE and Amble EBI. So, I think it's gone too far again. Yeah, okay. So then, after the development of tools and resources, coming now, I think, to the core of what BovReg is. The core is more or less, on the top you see this DNA chain, and what information is in this DNA chain. What do we make of it? And in the first part of the project, or this talk here, I will introduce all the different layers of information we put on top of this. And there, I think at the beginning, I had the recommendation from Michel Georges to have put this into the hands of one team, or mostly one team. And I think it was a very good recommendation to give this to our GIGA team, because there we have everything in one hand and very standardized way. So we have the transcriptome, the chromatin access. Also, we worked on the methylome. We also added interacting regulatory regions and even went to mobile genomic elements, which all might be found in the genome sequence. So now we have the feature that we have specific we have the same biological sample which went to one laboratory, and we have everything there done in one laboratory, and we have there the total RNA, we have the mRNA, we have the microRNA, we use the Oxford nanopore sequencing, and all this is done on a single sample in the same laboratory, on the same conditions. Also, the same is true for ATAC-seq and ChIP-seq. We also did, the team in Edinburgh, they did the CAGE also on the same isolated sample. And also for the whole genome methylation and RBS sequencing, this was distributed in the teams of FBN and INRA-A. So I think one of the really special points about BovReg is that we have a very, very standardized and consistent way how we collected all this information on top of each other for the same biological sample, taken at the very same day from the very same tissue. So this, first of all, will be definitely there will be an extensive talk from Gabriel, and I think Gabriel, I'm quite confident you could spend the entire day talking about the data. You will have to restrict yourself to 30 minutes. It's a challenge, I know. And we also have the opportunity, because we are working on this with the cell lines, we can also even compare this with the respective cell lines and compare how much they really represent what we find in the tissue. Unfortunately, we don't have the time to do everything we also did on this point. For example, we explored further the non-coding part of the transcriptome, circular RNA. A special thanks to Annie Robic, she might be in the audience. We did single nuclei sequencing with Doreen, and we also did 3D interaction experiments at INRA-A with Dominique. So we will hear more about the essential parts, the first parts, as I already mentioned, from Gabriel and Ling Tang. It's really a challenge to operate this device. Okay, then also what we did is, how does this match with other species? On the one hand, the team in Edinburgh, they compared how does the transcription start site match across other species, particularly also the focus on ruminants. And this paper was already published by Mazdak Salvati this year, so you can have a look at the paper, looking at the specifics of how the bovine genome is distinct from, for example, the human genome. And then the other part is comparative analysis annotation through conservation of sequence. And this is an extrapolation which was conducted at CRG with two specific approaches, and Björn will talk about this also in a minute, so that we have the comparison sequence-based regarding the functional annotation. And finally, same. See, yeah, it's the same box. And finally, on top of this, we thought that epigenetic sensitivity might be something which would be worth mapping on the genome. So where are sites which are particularly sensitive to environmental challenges? Where does the genome react to environment? And maybe even establish a memory of this environmental challenge. So we used in vitro produced embryos as models, and on the one hand subjected them to heat stress and looked how would this affect the transcriptome, the methylome, and the chromatin excess by ATAC-seq sequencing. And on the other hand, we had this heat stress model where sires which had been subjected to heat stress. There we collected semen which was collected at heat stress and at normal temperatures, and we followed up the embryos that were produced from this semen, and these embryos then were also monitored for potential impacts on the methylome and on transcription and chromatin accessibility, to see whether we even might see one intergenerational or transgenerational effects of this challenge. Then we had one last piece where we had used the functional annotation itself to integrate data with zootechnical traits, to integrate data for biologically informed breeding. And for this we added the molecular phenotype mapping and the well, let's say conventional GBUS mapping to our collection of assays. For this purpose, we, on the one hand, as I mentioned, we used the different layers of phenotypes, and what is maybe also specific to BovReg, for all those we had whole genome association studies with S&P ChIP-seq genotypes which have been imputed to whole genome sequence, all in the same standardized way which we had agreed upon in the beginning of the project. For the zootechnical QTLs, we had looked, for example, at growth traits. We looked at milk and reproduction. This is something which was done in very close collaboration with the 1000 Bulls Genome Project led by the groups in Australia. We looked at udder health, at feed efficiency and feed intake. And for this, most of the times we use large commercial population with existing background data, which we were able to feed into BovReg. For the metabolites, we use blood plasma metabolites with NMR or MSMS monitoring. We had monitored about 800 different metabolites and from 4 different populations. And for the expression EQTLs, we had 8 population, 8 tissues or cell samples. We used Nextflow transcriptome annotation with DSL2, something which we might hear more about tomorrow. And for this, we will have very extensive talks also tomorrow on the different kinds of phenotypes with all the information about where positions of particular interest might be in the bovine genome. So then when you put everything together on top of each other, so on the one hand you have the what we would say classical functional annotation, then the epigenetic sensitivity, and then the GBUS driven, so association driven regions which prioritize specific areas of the genome. Then in the end probably you would like to know which SNPs then to prioritize, because this is the current challenge we face in many applications. On the one hand for genome editing, but particularly also for which SNPs to include in our genomic predictions. So the challenge is you have a wealth of data, you have hundreds and thousands of SNPs which are highlighted in one or the other assay, which are the ones to use. And for this, we had a specific work package, so the challenge first is how do we prioritize the variants for functional validation and also for input into a genomic selection. For this, we use more or less generally two different approaches. The one on the left hand, the STIP pipeline, which was developed at INRAE with Dominique Rochard. They took all the existing SNPs in the database. They looked which of the SNPs might potentially affect if they are polymorphic, might disrupt transcription factor binding sites, and then which of those positions gave a hit or are coincident with a hit in our different GWAS and annotation assays. And finally, then we needed, because for function validation you need cell assays, we took advantage of all the data we had from our cell characterization experiments. We had the condition that these potential target SNPs, there the relevant target gene has to be expressed in the cell line, and also the transcription factor itself has to be expressed in the cell line. So then we come up with these little red dots there, which would be interesting for functional validation in cell models. On the other hand, and this is on very specific singular areas, whereas the TWAS SNPs, the TWAS approach, which was developed at Edinburgh, and which we'll hear more about tomorrow, this used a more wide approach by integrating expression data with GWAS data and coming up, filtering particular genes of interest and respective SNPs, which would then be relevant to put into genomic prediction evaluations and might be also contribute to prioritization of SNPs for validation in functional assays. For the functional assays, on the one hand, we had the functional validation of regulatory variants. For this, we used our 3 cell models, which we deeply characterized, the MDBK, the MACTs, and the F3 cells. On the one hand, we used reporter gene assays for the promoter variants, and on the other hand, we also did a lot of CRISPR editing of those cell lines, and then subsequently characterize those cell lines if there was an effect on the expression of the target gene, whether it had a global effect on expression by RNA-seq, and we also went further to functional cell assays, like, for example, energy metabolism or growth features. And on the other hand, we had this validation of the 3D interactions, also with the cell model, with our MDBK cells and the 3C approach. And even Hiroaki was so bold to include environmental effects, so to see whether this 3D structure holds also across different environments. You'll hear more about this from Hiroaki tomorrow. And then, in the end, I mean, this is all nice to have, but our stakeholders usually they ask for what is the benefit, what is the payment, and the payment is, in the end, to have an improved genomic selection informed by biology. And for this purpose, in work package 7, particularly, people developed models for biology-driven genomic selection. And here, on purpose, I particularly put Eurofang, because there was a working group and a very good collaboration between the different Horizon 2020 funded projects, because in the end, when you have the SNP and when you have the potential effects of those SNPs, it's more or less the same if you work on cattle, pig, chicken, or even if you work on plants, if you go a step further. So there were really a lot of joint efforts across the Horizon 2020 projects. And for establishing the models, they took all the functional annotation information we had from BovReg, but also they went into the literature and validated these genomic prediction models within large, but also particularly within small populations, because this was one of the objectives we particularly had for BovReg, that these efforts should not just benefit to the large populations, would even have the benefit like Holstein or Angus, but it should also benefit small populations like, for example, Jerseys. You will hear about this tomorrow in this respective session. And then I think this is also something which is special to BovReg. I already mentioned it. We looked at outreach and social impact, and there we looked at ethics and social issues. So on the one hand, we looked at the social contact of livestock genomics. It's an ethic. The colleagues performed an ethic analysis and also established stakeholder frameworks, because frequently, at least in our societies, we see that people become more and more reluctant regarding science and new technologies. So for us, it was very important to proactively look at potential perceptions, potential issues, which most of the times, I think we, as our researchers in the field, we don't really think about it. And it was, I think, a good opportunity we took, that we had the opportunity to have Curran and his team to look at these matters. And then we also had our team from Edinburgh, Donald Bruce. He had this Demox game, which was used then to engage the public. So people we are not really stakeholders or might not be from our community, to bring them into a discussion. And all the on-site participants, now they have the advantage to have one of these boxes of the Demox game, which they can take home. If there are people in the audience online, they can give us a note, and the material will also be available then for electronic sending. You just don't have the nice printout and the nice boxes. Yeah, we have two presentations on this matter tomorrow. The other then demand which all EU projects have to fulfill is communication, training, and particular for us it was clustering across the other Horizon 2020 funded projects. I think that's quite a common feature which is often asked by the EU to parallel funded projects, but I think there are few ones who achieved this to the same extent as we did with our Horizon 2020 FANG projects. So we were asked to cluster across the project. These are the projects from the first stage, which were Aquafang and Gene Switch, which have already ended. But there was also the second part of the Horizon 2020 call with the projects Rumigen, Geronimo, and Holoruminant. And we all now form this cluster of Eurofang and had a lot of joint activities, which some of them I will talk about in the next slides. So the first thing, we clustered across the projects. For the first 3 projects, Aquafang, Gene Switch, and BovReg, we established a common roadmap to see that there are no duplicates, that we make the most of the potential synergies. And it had this roadmap as a living document for tailoring and readapting the project during the time course of its lifetime. Then we had, for example, common Slack channels, for example, for the bioinformatics group. There's one common group who interacted on the genomic predictions. So there's a lot of plugins for the other Horizon 20 projects and also other FANG projects to interact with this Eurofang umbrella. We did a lot of workshops, training courses, and methods for wet lab bioinformatics and genomic prediction models. So, for example, already right in the beginning, we had this novel genotyping solutions course hosted by ETH. So it was a fully online course. It was during the time of COVID. And I think it was one of the first examples that such a workshop could really nicely work. We had courses on the use of Nextflow and NF-Core. We had a nice wet lab course at the premises of the University of Liège. And quite recently, we had also a training course on the methodology of biology-informed selection at Aarhus. Yes? Limoges. University of Limoges. Did I mention? Oh yeah, that's wrong. Okay, sorry. I have to correct myself. Definitely, the ATAC course was hosted in Limoges, and I think they even made a tour trip to visit the Limousin cattle there. For all these courses, we put together materials. So look at the BovReg webpage, look at our YouTube. Lots of this material can be downloaded. You can have, if you need material for the training courses, the contacts are given there. So also this conference will be, as I mentioned, fully recorded and will be available afterwards. So there's a lot of material as a legacy to the European and also global FANG community and beyond. And as I mentioned, you might be, even if you're not interested in FANG, even if you're not from the animal science community, there's a lot of material that you could use for your daily work. Oops. Yeah, then we also organized quite a lot of meetings together with the other Horizon 2020 projects. For example, the EAP meeting in 2021, which was still under the conditions of COVID in Davos. We had the large World Congress, also hosted by one of our project partners from Wageningen. And then quite recently, we had the EAP meeting in Lyon. And I think it was really a nice collaboration with the Horizon 2020 project, on top of all the other Congress participations we had, which go way beyond what I can list here in this short presentation. And I think most important and I think quite remarkable achievement is that we were able to establish the Eurofang research infrastructure based on this Eurofang umbrella projects. BovReg was partner, and until the mid of last year, I was the coordinator of this Eurofang research infrastructure, and it really achieved that the Horizon 2020 projects, they don't just end, which is a common, quite sad feature in many projects, that communities come together and then when the project ends, everybody dissolves and nothing follows up. But we were bold, and on the recommendation also from people from outside submitted this call to establish this research infrastructure, and we're quite successful. So within the Horizon Europe program, from 2023 to 2025, we will have this infrastructure which has the aim to establish an infrastructure to facilitate research within the S3 roadmap, so to really achieve sustainability and long-term support of our national and international funding bodies, to have one coordinated umbrella where we could act as a large community. This would be analogous to things like nanotechnologies, like atom energy and these things. So it's, I think, a really good achievement that we had, that our community does not dissolve, but has a structure and focus point how to move forward. And we will hear more about this also tomorrow in the talk from Klaus Wimmers. So what are the special features then of BovReg? So first of all, I think, to summarize, I think one of the special features is that we have a functional genome annotation of the Bostaurus genome, and it's a very comprehensive catalog because we have a huge diversity of biological samples across different environments, across different ages, across different breeds. So we have a really large diversity of Bostaurus. And then, as I already mentioned, we have a very rich portfolio of assays from the very, very same biological samples. So we have, for some of the samples, all of the samples, we have total RNA, microRNA, mRNA, cage, LNC RNA, circular RNA, and ONT RNA. And for some of the samples, we even have single nucleus RNA-seq samples. So all on the very same bits of tissue which has been collected. And on the other hand, we also have for all of the samples the classical CHIP and ADAC-seq assays. We have for all of them we have RBS data, and for the collection for which we have single nucleate data, we also have whole genome methylation sequence data. So what also makes BovReg special is that we have this philosophy of a reusable, interoperable, standardized data analysis pipelines, this NF core philosophy. I think this is something which was really introduced by BovReg into our community. We create an awareness for the limits of the in vitro cell line models. You will hear more about this in a few minutes. We really functionally validate that some regulatory variants have an impact. This is something which normally people stop at association, but we really want to achieve validation by demonstration with really wet lab experiments. We explore the drivers for the conventional and molecular phenotype diversity in large commercial population with S&P chip genotyping data. And as I already mentioned several times, we consider societal perception, including genomic information for cattle breeding. And last but definitely not least, what makes BovReg special is an extremely motivated team of highly skilled international scientists, where we also establish a lot of friendship between our colleagues. I think this is probably the most specific about BovReg. And with this, I hope to see all these colleagues which are here depicted and contribute in future networks and joint projects. And at this stage, already quite early, because I know tomorrow there will be a wrap-up, but most of the times in conferences, wrap-ups suffer from some dilution of participants due to flight schedules and everything. So at this stage, I would like to make the big thank you all for this excellent collaboration across so many partners. And I really apologize for all those non-named colleagues in the background, which sometimes are essential to our work, but sometimes really we fail to name them by person, and explicitly want to thank all of them for their input, particularly also in the times of COVID, which would not have been possible to have this project without their contribution. And here it's only the main, or what sometimes are the most visible colleagues. So there is cohorts of people in the back who also contribute. And with this, with the logos of all the partners, I would like to conclude. Thank you for your attention. And if there are questions already here at this stage, feel free to ask. Yeah. 1 minute, we take the microphone and then. 1 minute, we are checking the... Okay. If you just take your name and... Yeah.
Sure. James Coltis, Iowa State University. Thank you for the very interesting talk. For the folks in the room, they're familiar with this. I notice from a U.S. perspective, in particular, the work on reproduction and integrating with stress physiology with the heat stress work. And I'm curious, for the European Union funded projects, is there becoming a lot more emphasis on integrating with the other animal sciences and extending and showing the opportunity to leverage your FANG findings across the animal sciences? This has become a big emphasis in the US and a big emphasis of projects moving forward. And so I'm kind of curious the European perspective on that to kind of help our colleagues across the broader animal sciences to leverage your findings.
Yeah, I know that, for example, already in Rumigen, one of the follow-up projects, there is some follow-up of this environmental impact. But for example, for the Eurofang research infrastructure, we also will we have a specific work package on integrating phenotyping, which then needs for example, animal nutritionists, veterinarians, and particularly the veterinarians. You know that I myself moved position, and this will definitely one of my missions to bring together the two communities, because from experience I see that we have still our bubbles, but we could tremendously benefit. For example, you mentioned the heat stress, also for example the single cell work where immunologists from an entirely different perspective working on vaccines would definitely benefit from cross-fostering, yes.
Maybe just one other comment. There's obviously a lot of experimental work that can be done there, but also some annotations that would be pulled back from collaborating across all of the different aspects of biology as well. So I think there's a lot that could be done there to help link genotype to phenotype by reaching out to the different disciplines.
Yeah, I think we have to raise awareness, and this is definitely something we will be working on. And I would assume that will meet also the expectations from the EU, because what they want to see is, I think, in the end, output for stakeholders, and output for stakeholders is definitely always much more straightforward if you integrate. Other questions from the audience or online? No questions? Okay, I think then we are even a bit ahead of time, but this I think is not too bad, because we can then carry on. We want to move forward to the next session. Initially, we said that speakers would have about 15 minutes, including discussion, except some of the main large fields of discussion. So I would ask then Carol to take over for the chairing of the next session.